Welcome everybody to the California Department of Fish and Wildlife's Conservation Lecture Series. My name is Nicole Russell. I am the Conservation Science Specialist for the Science Institute, and together with my colleague Whitney Albright, who is also on the line today, we run this series. And before we get started here, I just have a few reminders. First, everyone is muted to preserve the audio quality for our recording. So indeed, the talk is being recorded if you want to catch it again later. And our format today is to show PowerPoint, and that will be followed by a dedicated question and answer session. So if you have any questions, please do type them into the chat box, and Whitney and I will be monitoring, and I'll be fielding those questions on behalf of our speaker. Our speaker today is a wildlife forensic specialist at CDFW's Wildlife Forensic Laboratory, so one of our own. And not only that, but Kelly Carruthers, I should say, was the recipient of an Employee Excellence Award this past year for her work in the category for scientific excellence. So we're really excited to have her. And before joining DFW in 2016, Kelly received her bachelor's degree in biochemistry and molecular biology and master's degree in forensic science, both from UC Davis, where she focused on forensic DNA analysis with an emphasis on the population genetics of Native Americans in North America in an effort to improve random match probability calculations in forensic casework. She currently dedicates most of her time to mitochondrial DNA analysis related to ivory and wildlife trafficking. So please give a virtual welcome to Kelly. And with that, I will hand it over to her.
All right. Thank you, Nicole, for the kind intro. Okay, so thank you to everyone for attending my talk. I hope you find it interesting, maybe a little out of the normal. And so, yeah, I guess we can just dive in. Elephants, Ivory, and Trafficking, how the CDFW Forensics Lab is helping to combat the illegal ivory trade. And I'm Kelly Carrothers. Okay, so just a quick presentation overview so you guys know what you're getting into. I'm just going to go over briefly what wildlife trafficking is, and then what's happening with the ivory trafficking and the ivory trade globally, go through some of the trends that we're seeing, and we'll talk a little bit about California's AB 96, which was the ivory ban that was enacted in 2016, and then what the Forensics Lab is doing in terms of genetics work, some geolocation work with African elephants, and then some other non-genetic related projects that the Forensics Lab is doing to support the AB 96 bill. So global wildlife trafficking. Wildlife trafficking, wildlife trade, it's essentially whenever people sell or exchange wild animal and plant resources. So it can involve live animals and plants, or really any kind of wild animal and plant product. And what's driving the trade is the end consumer, who has a need or desire for wildlife products, whether that be food, construction, clothing, traditional medicines, high-end fashion, or really any other byproduct from an animal or a plant. Illegal poaching and wildlife trafficking is the fourth largest transcontinental crime, and it's estimated that it's worth an estimated $20 billion each year. And again, it's fueled by growing markets, primarily in the US and in China. Perhaps the most obvious problem associated with wildlife trade is that it causes an overexploitation of particular species to the point where that survival of the species is put at risk. Overexploitation can lead to larger problems with the whole web of life, as non-trafficked species depend on the presence of trafficked species to survive, essentially. And so wildlife trade can also cause indirect harm through introducing invasive species, which can prey on or compete with native species in a particular habitat. It can cause the incidental killing of non-target species. And a good example of that would be, dolphins that get caught up in nets for the shark fishermen. So just to provide a glimpse of the scale of wildlife trafficking, or basically what's being reported within a year's worth of wildlife trafficking, there's been recorded over 100 million tons of fish or fish species, 1.5 million live birds, 440,000 tons of medicinal plants. Again, that's just within 1 year of international trade. So it's a huge industry. So how does this relate to elephants and ivory? Again, habitat loss, poaching are both driving down populations of African and Asian elephants. And there's really two main regions in Africa where this ivory is coming from. African forest elephant ivory over in the west central area of Africa, and African savanna elephant ivory from Eastern Africa. It's estimated every year 96 elephants are killed every day, and 50,000 elephants are estimated to be poached every year. Overall populations of African elephants in particular have decreased by about 100,000, which is a pretty significant number. A driving force behind a lot of this poaching is just the huge jump in value that ivory saw. Back in the 70s, ivory was priced at $2.50 for a pound. In the early 2000s, when the price of ivory was peaking, ivory was valued at over $90 a pound. And so as you can imagine, these large bull elephants are being targeted because they have more ivory per elephant. And so it's leaving herds with skewed sex ratios and just an overall loss of genetic diversity. And back to the web of life, elephants aren't the only populations that are seeing a decrease in the genetic diversity. They're a primary seed disperser for a lot of the large trees in Africa. And so a lot of the forests in Africa are seeing a decrease in the numbers and the amounts of trees that are growing up there. So cutting down these populations is having a huge effect. These are some numbers that I found off of the CITES website that I thought were really interesting. I went ahead and placed a circle with a slash wherever a recent ivory ban had been enacted. In the United States, the Endangered Species Act was updated in 2016 by Barack Obama to include a near total ban on elephant ivory trade in the United States. Thailand and China also placed pretty recent bans on their domestic markets for the sale and import of elephant ivory. The US and China are two of the top 4 ivory markets in the world. Vietnam has recently become the world's leading destination for illegal ivory, surpassing China, and it's implicated in about 22% of the total large-scale seizures. And a large-scale seizure is any seizure of 500 kilograms of ivory or more. And so what's really interesting when you look at these numbers is, countries like Singapore, Vietnam, China, they don't have a lot of number of seizures, but the amount of ivory per seizure is pretty large. And if you just look at Vietnam in 2019 with over 10,000 kilograms of ivory, when one elephant has about 6.7 kilograms of ivory, that comes out to about 1,500 elephants in 2019, which doesn't seem like a lot, I guess, in the grand scheme of things. But when you think about, these are just the reported seizures, and it's estimated only 10% or so of seizures get reported. Suddenly the numbers really skyrocket. The good news is in 2019, there were 946 seizures that were reported by customs. In 2020, there were only 466. So the number of reported seizures dropped drastically by about 50%. Unfortunately, according to the Environmental Investigation Agency, only 14% of those seizures resulted in a conviction of any kind, and unfortunately, only a fraction of these large importations are being reported and are actually being seized. So a lot of ivory moving around the world. So as I mentioned before, in 2016, Barack Obama updated the Endangered Species Act in the United States to include elephant ivory. This is just an overview of what some of the other states are doing. California houses 2 of the 3 largest ivory markets in the United States. New York is the top ivory importer, and so they've placed a ban in effect for elephants and mammoths. But it's nice to see other states kind of jumping on board. Okay, so California's efforts to ban the ivory trade. San Francisco and LA are 2 of the top 3 markets for ivory in the US, and most of the ivory that comes in is either through airports or shipping ports. In 2015, Assembly Speaker Tony Atkins passed Assembly Bill 96, which prohibits a person from purchasing, selling, offering for sale, possessing with intent to sell, or importing with the intent to sell ivory or rhinoceros horn, with some exceptions. And there are 10 species included in the AB 96 bill. So the 4 that I'm going to focus on today are African elephant, Asian elephant, mammoth, and mastodon, which all fall under the order of Proboscidea, which is a word that I'll be using quite a bit today. And it just means elephants and their extinct relatives. But AB 96 also includes walrus, hippo, narwhal, killer whale, sperm whale, and warthog. So why does CDFW care, and what are we doing to help? Well, the forensics lab was tasked with finding a way to utilize small amounts of DNA within a piece of ivory to figure out where that ivory came from or what species that ivory originated from. Not only do we need to identify what species the ivory came from, but we need to be able to exclude elephant ivory substitutes like plastics and resins and bone, or ivory that's commonly used to fraudulently traffic elephant ivory, such as mammoth and mastodon. Mammoth and mastodon are not protected by CITES, and so traffickers will often intentionally misidentify or lie about the ivory that they're importing. So they'll say that the ivory is from mammoth or mastodon, when really it's an African elephant ivory. And that's just to get around the CITES laws. AB 96 closes that loophole though, so they can't say it's mammoth or mastodon. All right, so how can we make a taxonomic identification from ivory using DNA? Well, the simple answer is we're going to look at the mitochondrial DNA to make that species or that taxonomic identification. But before I talk about, get too far down the genetic rabbit hole, I just wanted to take a moment to talk about our sample collection and the types of samples that we've been able to analyze at the lab. So down at the bottom right, I've got a chart there of all of the ivory that the lab has looked at the mitochondrial DNA of. So, and I want to make a note that African elephant includes 2 different species. So it's actually a genus call. It includes the African forest elephant and the African savanna elephant. Asian elephant is a species. However, our 31 Asian elephant samples included 2 subspecies of Asian elephant, the Indian elephant and the Sri Lankan elephant. Mammoth is also a genus call, and it includes the Colombian mammoth, the woolly mammoth, and the Channel Island pygmy mammoth, which I'll talk about in a second. And then mastodon is also a genus call, and it includes the well-known American mastodon, as well as a brand new species of mastodon that was just discovered in 2019 called the Pacific mastodon. And that picture up on the left of my colleague, Ashley Spicer, and I, we were at the Western Science Center talking to Alton Dooley, who was the researcher that discovered the new species of mastodon. He was able to give us 4 different Pacific mastodon samples, which are included in the table below. Pacific mastodons existed in North America all the way south down to Costa Rica during the Pleistocene era, whereas the woolly mammoths were in the Arctic region. So again, totally new species. And we're actually the first lab to do any genetic work with the Pacific mastodon. So pat on our shoulder for that. The picture at the bottom left was a Toss the Tusk event we did at the LA Zoo, where we were able to collect over 100 pieces of ivory from the public, just people that wanted to donate their ivory because they didn't have a use for it. Really successful event. The picture up at the top is from La Brea tar pits. Unfortunately, because of the tar, we weren't able to recover any genetic material from the mammoths and mastodons we sampled at La Brea tar pits, but it was still a really fun and educating experience. And they have a lot of mammoth and mastodon samples, which is why we wanted to go there to sample. And then this tusk on the right, really interesting. This is just, it wasn't a sample collection trip, but it was just an example of some of the really interesting samples we've come across during the course of this project. We had a researcher down off of the Channel Islands of California, mapping the bottom of the seafloor, and his sonar system detected a tusk-like shape at the bottom of the ocean. They dredged up the tusk, and this is a cross-section of the tusk that they sent to us for a species identification. Ended up being a Channel Island pygmy mammoth, which is a very special dwarfed elephant that lived only on the Channel Islands during the Pleistocene era. Super interesting. And it was dated to be at least 150,000 years old, even cooler. So just some of the really interesting samples that we've had to work with. So once we have our samples and have our ivory, the biggest issue is getting DNA out of that ivory. So, there's a couple considerations when you're dealing with ivory is, the age of the item. So for example, this 150,000-year-old tusk, DNA isn't going to be super fresh on that piece of ivory. It's going to be really broken down. It's going to be segmented. It's going to be degraded. Ivory requires the demineralization of all of the calcium ions that are present. Calcium ions bind up the DNA, and so you've got to get rid of the calcium before you can get to the DNA. That process is time consuming. It requires harsh chemicals. It doesn't always work. There's the ever presence of human contamination. Humans are constantly touching these items, and so it's an important consideration to take into effect. You don't want to be extracting human DNA. And for things, not necessarily ivory, but leathers and hides of elephants, there's the unknown previous treatment aspect. So heats, dyes, tanning processes all disrupt the DNA. And because of all of these challenges, that's why we're using mitochondrial DNA to make the species or the genus call rather than nuclear DNA. So how do we recover DNA from ivory? We use a handheld Dremel tool. We fondly call this method the Smittle sanding method, after one of our wildlife trafficking officers, Garrett Smittle, who came up with the idea. We collect 150 milligrams about approximately a fine powder. And I just want to make a note that ancient DNA work, so mammoth and mastodon work, is done in a completely separate facility, different hood, just to make sure that the contemporary DNA isn't contaminating the ancient DNA samples. The powder then undergoes a 24-hour demineralization step, and then it's extracted. So the whole process of the demineralization and extraction is about 26 hours. So it's relatively not that slow, it's pretty quick. Once extracted, DNA is analyzed on our Quant Studio instrument, picture of it on the left. Essentially what's happening in that instrument, DNA is undergoing what's called a polymerase chain reaction. So it's being amplified millions of times. And this allows the instrument to be able to detect the DNA. Once the DNA is amplified, it's then melted using just a high temperature. And that melt peak that is created is essentially associated to a particular species or a particular genus. And that process is called the multiplex high resolution melt. So multiplex high resolution melt, it's used quite often in forensic science, and it's based on the idea of SNPs, or single nucleotide polymorphisms, which are just base pair changes of your DNA sequence. So your A, T, G's, and C's can switch, and they're different between species. The multiplex high resolution melt can be used for phenotyping. So the H-IRISplex is a HRM assay that looks at different SNPs in eye color. You can tell body fluids, and you can do species ID. So things like herbs, plants, domestic animals, tigers, and now proboscideans. Common geolocation or biogeographical ancestry site is ancestry.com. It also uses SNPs to look at your ancestry. So it's a very common method. Multiplex high resolution melt of proboscidean ivory. So this is what was developed and validated in-house at the forensics lab. And we're essentially detecting multiple SNPs at once. We're detecting 16 SNPs in one run using a combination of 2 genetic sites to make our species or taxonomic identification. We're looking at a 16S and a cytochrome B gene. Both are 2 universal genes on the mitochondria. And the primers for those sites, as I said, were developed and validated in-house. So they're novel. This technique is highly sensitive. It has a 0.5 picogram limit of detection. And just for reference, a human cell, one cell, has 6 picograms of DNA in it. So we're talking about a fraction of a cell is what this assay can detect. So it's very ideal for short and degraded DNA sequences, or such as, those mammoth and mastodon samples that I talked about earlier. It's quick, it's simple, it's reproducible, and it's cost effective. So this is an example of the output after a run on our Quant Studio instrument. So the 2 genetic sites that we're looking at, 16S and cytochrome B, are on the x-axis there. There's 3 peaks, S1 through S3, that are associated with 16S, and then there's 4 peaks at site B, C1 through C4, which are associated with the site B sequence. And each of the 4 species, African, Asian, mammoth, and mastodon, have a unique combination of S and C peaks. And those are essentially called composite melt profiles. So example composite melt profiles are on the right. S1C2 would be a P2 composite melt profile, and that's associated with an Asian elephant. And I do want to point out that these primers for both of these genetic sites were extensively validated and developed so that human DNA does not amplify. So there's no human being amplified. And this is just a figure from the paper showing the 3 composite melt profiles we obtained for Asian elephants, the 5 composite melt profiles we obtained for African elephants, and our mammoth and our mastodon composite melt profile. And then I'm not going to spend too much time on this slide. There's a lot going on. This is basically my proof of concept to prove that what I'm amplifying and what's melting is not human. It's not the result of environmental contamination. It's truly a proboscidean specific DNA sequence. And what's happening on the right here is this zoom up picture of a strand of DNA. And so our G's and our C's are bound by 3 hydrogen bonds here. Our A's and our T's are only bound by 2 hydrogen bonds. So essentially it requires more heat and more energy to break apart a GC bound pair than it takes to break apart an A and T bound pair. So sequences that have higher GC content are going to be shifted right on the x-axis. So if we go up here, so these C2 sequences here have more GC content in their DNA sequence than these C1 peaks. We know that because the takes more temperature to break the bonds of the C2 peak. All right, so now that we can tell if a piece of ivory is from an African elephant, how do we determine where that piece of ivory came from in Africa? Well, we're taking a 2-pronged approach using both mitochondrial and nuclear DNA. So just a quick mitochondrial DNA overview before I get too far down this rabbit hole. Kind of talked about it before, but mitochondrial DNA, it's maternally inherited. So you have the exact same mitochondrial DNA as your mother and as your mother's mother and as your mother's mother's mother. So it's linearly inherited. There's no recombination going on. And it's extremely easy to track that way, if you will. And it's really easy to make a species or any kind of taxonomic identification based on mitochondrial DNA because there's not a lot of variation going on. Nuclear DNA comes from both of your parents, 50% from your mom, 50% from your dad. It's much larger than mitochondrial DNA, and it's essentially unique to each individual. So it can be used for individual identification. It can be used for geolocation. It can track migration events. It can be used in forensic science. It can be used to create random match probabilities in casework. So did this sample come from the suspect kind of thing? Okay, so mitochondrial DNA in African elephants. So there's 2 species of African elephants. There's the African forest elephant and the African savanna elephant. The dark green region on this map shows where the African forest elephants live. And then the tan region on the map shows where the African savanna elephants live. And then this light green zone is what's thought to be a transition zone between the two. There's 2 main haplogroups present in African elephants, the F clade, which is observed in both forest and savanna elephants, and the S clade, which is observed only in savanna elephants. And so these orange crosses on the map basically are indicating where the S clade haplogroup is present. And since the S clade is only present in savanna elephants, we essentially know that there's savanna elephants in the tan region. And this graph on the bottom right here is just showing that, the more likely you are to have an S clade haplogroup, the more likely your nuclear genotype is going to correspond to an African savanna elephant. So this is just kind of showing where these 2 clades coexist. Now, what I've been able to do is, as I mentioned before, we've got our 5 African elephant melt profiles that we've observed in our population at the lab. And I've been able we've color coordinated here. So our P3 composite melt profile is seen in primarily Uganda, Cameroon, and the DRC. And you can see that region on the map there. And so I've been able to assign a very broad geographic region to each of our African elephant composite melt profiles, which is giving me some information. It's not incredibly helpful in terms of pinpointing a country, but it gives me an idea of where the ivory is coming from in a broad sense. If I overlay what I've found with my composite melt profiles with what other researchers have done and found with the S and F clade haplogroups, I can make a few more inferences about my data. So for example, it does look like the P3 and P5 African elephant composite melt profiles are in fact African forest elephants. And our P9 composite melt profile African elephants are savanna elephants. And this P6 group is an F clade, so it could have a bit of hybridization going on between the forest and the savanna elephants. But it does look like it's primarily savanna, an F clade savanna elephant population there. And I do want to point out this region here, Garmaba, which is in the DRC, is a region known for having a very high hybridization rate between the two elephants species. That's really the only region where hybridization is occurring, which was a surprising find to a lot of researchers. So we can currently associate specific regions in Africa to each of the composite melt profiles we obtain, making both the taxonomic identification and geographic assignment in a single run. So there's some issues with using mitochondrial DNA for establishing geographic provenance. Most obvious is that mitochondrial DNA is maternally inherited and elephant populations are male dispersed. Mitochondrial DNA also relies on a reference map, and it assumes that researchers went through and sequenced a bunch of samples from known regions in Africa. So taking the first two bulletins into consideration, since those are the two we can kind of control for, we're partnering with Dr. Glendon Parker over at UC Davis Department of Environmental Toxicology, and he's going to see if he's doing a pilot project to see if he can estimate the sex on a piece of ivory using proteomic techniques rather than genomic techniques. And I won't really speak to what proteomics is, it's above my head a bit, but that's why we're working with Dr. Parker. We're looking at amelogenin, which is a protein that is primarily codes for enamel in teeth, so it's a perfect candidate for ivory. And I guess the reason that we're really wondering about sex estimation is just due to elephant behavior. So males can travel up to 120 miles in one day, which females, opposite of that, don't travel a lot, or they'll travel and they'll come back to where they're originally from. And so I drew some circles, just some random circles on this picture map of 120-mile radius. And so you can see after a couple of days of traveling, a male elephant can essentially get out of that region that their mitochondrial DNA is putting them in. And they're just traveling around, looking for mates. So essentially, if we have a male elephant or ivory from a male elephant, we can say, where that elephant came from, where its original herd was from, but not necessarily where that elephant was when the poaching took place. Whereas if we have a female elephant or ivory from a female elephant, we can say not only where that elephant came from, but likely where that poaching took place. So, being able to estimate sex on a piece of ivory could be hugely important when looking solely at mitochondrial DNA for geographic assignment. Since mitochondrial DNA only gives us a broad idea of geographic origin, we can also use nuclear DNA to get a precise idea of where the ivory came from. And so this is what we're working on now. We just started working with nuclear DNA to determine geographic origin. So same thing with mitochondrial DNA. Researchers have gone around Africa and created genetic profiles from distinct populations in Africa, looking at allele frequencies that are occurring in different populations. So this is very similar when you hear about the CODIS database in human forensics. We've got a bunch of allele frequencies, and then you're trying to match a particular unknown sample to this database to figure out, who did the crime, or what race the suspect was. So determining origin of a particular ivory sample involves looking for shared alleles between ivory and different elephant populations at 16 different genetic markers. And so nuclear DNA takes into account things like gene flow and migration patterns, which is required when you're looking at elephants, just because their pack structure and their various behaviors. So this picture on the left here is just an example of kind of how it works. So we've got our ivory sample, and we've got two different elephant populations, population A and B. If you look at the two lines under the ivory sample, you'll see that the top line is shared between both populations, but the bottom line is missing in B, and it's shared with A. So we would infer, just based on this one site right here, that this ivory sample came from population A. So we're working with Dr. Sam Wasser at the University of Washington to implement the 16 variable genetic sites into our ivory DNA workflow. Again, nuclear DNA provides just much greater precision than mitochondrial DNA. These maps on the right, there's a lot going on, but if we just look at Garmaba, this map here, these green crosses are the known reference population. So just dung samples that they found, they know where those dung samples came from. They were able to create genetic profiles from those samples. And we've got the true location of a known sample is this red cross over here. And then these blue circles are unknown samples that we're just trying to geolocate. And so based on the allele frequencies of the unknown sample and comparing those frequencies to the allele frequencies in our reference population, we can kind of narrow down where these unknown samples are coming from. And so this tight grouping of blue circles indicates that we're getting really close, very precise with our geolocation using nuclear DNA. And just for comparison, I copied and pasted my mitochondrial region map down here to just look at the regions that we're able to assign an ivory sample to. So much more precision is gained with nuclear DNA versus the mitochondrial DNA. On top of the precision, we are also able to resolve the two African elephant species. So we could resolve forest elephants from savanna elephants, which isn't too much of a legal concern. That's more just for interest's sake. We're also able to use statistics to do matching and exclusion. So if we've got, an ivory chess set with 32 pieces, we can say, hey, 6 of these pieces came from the same elephant. And going hand in hand with that, we can establish an accurate MNI, or minimum number of individuals. So if we've got a case with a bunch of ivory, we can say, at least 4 African elephants were involved, or at least 10 African elephants were involved. So just a quick summary of what we're able to do now with mitochondrial and versus where we're going with nuclear is our MHRM assay is quick, it's easy to use, and it's good for processing a large number of samples. Works on all sample types, even highly degraded and old samples like mammoth and mastodon. It gives a taxonomic ID and a broad geographic assignment in one run. There's no associated statistics since mitochondrial DNA is linearly inherited. It doesn't take into account pack structure and social behaviors of elephants. It requires a sex estimation to improve accuracy. And it's essentially just a qualitative assessment. What is it? Roughly, where did it come from? Whereas nuclear DNA, it's expensive and it requires very specialized equipment. It's much harder to retrieve from ivory samples, just an overall lower success rate of retrieval, which is one of the big obstacles our lab is going to face with incorporating nuclear DNA into our workflow. Nuclear DNA provides a species identification, precise geographic assignment, and things like random match probability for use in casework scenarios. We can associate large number of statistics to nuclear DNA since it undergoes recombination. We can use the product rule essentially to look at genotype frequencies. It takes into account things like migration, gene flow, which is directly related to elephant pack structure and their social behaviors. It doesn't require knowing the sex of a piece of ivory. And so it's a very quantitative assessment. So that's where we're looking to go. To wrap up the DNA work, I just wanted to mention briefly some of the other work that the lab is doing. That's not DNA, that supports AB96. The big one is volume testing. So some exceptions to AB96 include provisions detailing the percentage of ivory by volume that qualifies as exempt. For example, AB96 doesn't apply to ivory that's part of a bona fide antique, and that is less than 5% by volume of the antique if the antique status is established by the owner or seller with historical documentation, and it's not to be less than 100 years old. So Ashley Spicer at the Forensics Lab has taken the lead on this project to develop a technique that non-destructively determines percent volume of ivory. So she's been working with Stephen Lucero at the UC Davis Teams Lab to essentially answer the question, does ivory percent volume meet the threshold? And so to just take you through the workflow real quick, we've got, for example, a statue here that has some percent ivory. So if you optical 3D laser scan that object and create models, you can essentially determine if that object is solid. So if there's no percent difference between your calculated and your measured volume, then you've got a solid object, and you can just do volume over volume to create percent volume. If your object has any sort of hollowness, things get really complicated really quick. And yeah, it's a workflow to try to figure out percent volume, and she's done a really good job of putting that in words that are understandable. And our scientific aide, Mark McClellan, is investigating the utility of a handheld XRF device for use in field screening. So he's looking at the calcium to phosphorus ratio of ivory. And what he's found is that if your calcium to phosphorus ratio is greater than 2 and less than 3, it indicates that the object you're scanning is probably ivory rather than bone or any kind of fake plastic shell. So this could really be useful in saving both officers and lab staff analysis time when we don't have to look at as many fakes. So really cool work by Mark. And I just wanted to give a shout out to all our collaborators and contributors that have helped on this project. Couldn't have done it without you. And with that, I'll take questions.
Wonderful. Thank you, Kelly. That was so interesting. Our first question actually is, how can I donate my ivory to you to help the forensic program? So I hope that person who asked is still on the line. So here's that information.
Yeah. Well, I hope that this last screen kind of helps with that. We're always open to taking donations or loans from anybody. If you just want to shoot me an email, that would be great. And we can set up a time for you to stop by either in person to drop the ivory off, or we could help you make a shipping label to ship that ivory to us. And then you can decide if you want to loan it to us or donate it to us and what kind of destructive testing you'll let us do on it. But we appreciate all donations. So thank you for asking.
Cool. Great. Okay. Another question, and these all came through anonymous. I'm not sure if that's an artifact of our setup here or what, but someone says, can you tell how old the ivory is? Do haplotypes change over time?
So the most common way of dating ivory is through radiocarbon dating. And we've got a contract in place with the Lawrence Livermore Laboratory to do our radiocarbon dating. It actually doesn't have anything to do with genetics. However, to answer your follow-up question, haplotypes do change over time. It doesn't take, there's not a lot of changes that happen, and it's got to be present over a very long period of time. But haplotypes aren't a reliable way to date a piece of ivory. Radiocarbon dating is.
Okay. Another testing related question. Someone asks, what happens to the ivory after testing? And I'm guessing based on what you said about the donations is it could be a number of different things.
Yeah, this is one of the most common questions that we get. But after ivory is tested, we pretty much just store it at our lab. We've got a couple of storage facilities throughout the state that are filling up quickly. But we try to loan out the ivory that we have to various institutions, either for educational purposes or for research purposes. As we start taking on casework for more states, hopefully in the future, we'll probably package up some of the ivory as a permanent loan to that state so that they can train their officers. The canine unit, we have a ivory detection canine unit that uses ivory to practice sniffing it out. So a lot of stuff happens to the ivory, but it pretty much just sits in our lab.
Okay, interesting. I had no idea. And it's great to hear that some of the ivory goes to expanding programs across country. So another person asks, what does the concentration of activity in the Zimbabwe area indicate? West Zimbabwe and northern Botswana of both species of elephant.
So I assume that you're talking about, go back to the map that I'm assuming you're referencing. There was a bit of a lack of sampling that took place in this area at the time of this research. And so that's why you're seeing a combination of both the orange crosses and the gray triangles. Just because Escalade wasn't detected in that region doesn't mean it's not there. It just means that either there was a small sampling size taking place, or I guess it could mean Escalade's not present, but from everything that I've read outside of this paper, it's more than likely just due to sample size.
Okay. Yeah, I was thinking about this map too. And if it would be more difficult for poachers to work in the forested areas versus the savannas. So if you would see that come out in your sampling at all.
Yeah, I don't know how much work poachers are willing to put in to get their elephants, but I would assume that forest territory would probably be more difficult to poach in than savanna, which is why you see higher numbers of savanna elephants maybe in the trade.
Yeah. Okay. So a question. What is a narwhal? And is that ivory the same as elephants? Google is your friend, no offense. But
Narwhals are the unicorns of the sea. They're the whales with the one tusk coming out of the front of their face. And there's only maybe 10,000 narwhals in the world, and they all kind of live around the Arctic Circle. So way up north, there's not a lot of them. And I believe it's 1 in every 500 narwhals actually has 2 tusks, which is
oh my gosh,
pretty interesting. Yeah.
Wow, I've never seen one like that before.
They're pretty rare.
Okay, another narwhal question. Somebody wonders if the narwhal tusk is hollow.
Oh, that's an interesting question, and it makes me wonder if you've seen a narwhal tusk, because they are actually hollow. They're hollow all the way through, which makes them, I think, easier to maneuver.
Oh, yeah.
For the narwhal.
Yeah, underwater, you don't want a big, heavy tusk, I guess. Interesting. Okay. So another person wonders if you work with zoos that have elephants, and I'm not sure what the context is, but that is an interesting question.
Yes, we partnered with a few zoos. So we've got San Diego Zoo has a bunch of African elephants that they've been sending us samples of. LA Zoo, they only have Asian elephants, but they've sent us samples from those elephants as well. We work with the San Francisco Zoo, which they have hippos and rhinoceros, but they don't have elephants. But we just started working with the Oakland Zoo, which has a few different African elephants as well.
Ah, interesting. Okay. Oh, here we have someone who actually is their name, Willetta, wonders how often does environmental contamination skew the results or otherwise interfere with DNA research? And it sounded like this is a pretty common issue.
Yeah, environmental contamination is potentially a very huge issue when you're working with samples that already have such little genetic material. So one of the biggest ways we avoid environmental contamination is through bleaching the surface of the ivory before we sample it. And then we'll sand off an outer layer of the ivory, discard that outer layer, and then go back with our Dremel tool and sand off an inner layer of the ivory. So that way, that's essentially the best we can do to ensure there's no contamination. And the only thing that's in the tube is ivory powder.
Ah, got it. Kind of reminds me of how they tell you when you get cantaloupes or something to wash the outside before you cut into it, because you can transfer, germs and things from the outside to the inside. Or you don't.
I haven't heard that about cantaloupes, but I think that's pretty good advice.
Yes. Please wash your melons, folks. Okay, well, I think I'm going to go ahead and ask this last question that came through. I think the answer is probably no, but someone asks, if I loan ivory to you, can I get it back undamaged?
Can you get it back undamaged? You could get it back undamaged. We wouldn't be able to do any testing on it. It would be, I guess we could 3D scan it for volume analysis. But if the item is pure ivory, probably won't be useful. So unfortunately, when you loan us ivory for genetic testing, there will be a little bit of damage, but we do work to minimize the damage. And a lot of the people that pick up their items that they loan to us can't even tell us where we sampled. Okay. So if you're worried about that, we can definitely help circumvent that issue.
Great. Well, it seems like you answered this next question that came through, which is the damage minimal at least? So that would be a resounding yes.
Yes, it's very minimal. I can give you contact information for people that have donated ivory and that haven't been able to detect where we've sanded. And if you're interested in pictures of things that we've sanded in the past, I'd be happy to supply those. But ultimately, we take loans as we respect it as your property, and we do our best to keep it intact.
Excellent. Okay, well, it doesn't look like there are any more questions from the audience, but I was curious about a couple of things, if you don't mind.
Sure.
Okay, so first, do you know, or does our department work at all with groups on the ground in Africa who are trying to combat poaching, or is that more of a different level?
Yeah, that's a different level. Some of our enforcement people might be able to answer that better. In terms of like lab work, we are working to establish some contacts with some labs in Africa that have samples, but we haven't gotten to that point yet. We're establishing connections with various labs in Africa.
Okay, cool. My other question, oh, I just forgot it. See if it'll come back to me. Sorry. Yeah, I forgot. Sorry. Maybe I'll shoot you an email later.
There you go.
Okay. Oh, let's see, Q&A. One more squeaking through. We'll call this the last one before we close out. Erica says, how many evidence samples are you testing and how many of those end up being fake?
That's an interesting question. So we've in the past had some issues getting permission to do genetic testing on casework samples. Most of our casework samples that we've tested have been after the fact, so after the case has been disposed, and then we're allowed to test items. The items that we have been allowed to test that were part of an active casework or case have been essentially all African elephant. There's been a few mammoth samples. I have yet to see an Asian elephant in casework, which aligns with what the federal lab is seeing as well. But predominantly African elephant ivory is being seen with very, very few fakes. So our trafficking or SOU officers are doing a really good job with determining what's fake and what's real before it gets submitted to the lab, which is appreciated.
Oh yeah, that's helpful. Filter it out.
Exactly.
Cool. Well, I just remembered my other question, which is if you had a wish list of what could be done to make your job a little easier, is there anything you can think of off the top of your head as far as like tools or collaborating with others, or more samples coming through, things like that?
Yeah, we're always looking for more samples. Can't have enough samples. Samples are what makes these projects happen. So we've done a pretty good job of establishing relationships with the zoos and research institutions within the state that are working with elephant samples. But if anybody has a contact of somebody that works with elephants, or somebody that has a connection to a lab in Africa that's doing elephant work, things like that, more of that would be extremely helpful, because establishing those connections is extremely time consuming and a lot of the times falls flat.
Oh, okay, good to know. Well, Science Institute is trying to help the rest of the department with its science work, so we'll definitely keep all of that in mind. All right. Okay. Well, thank you so much, Kelly. This was a really unusual and interesting talk, and thank you to all of the attendees who stayed on, asked some great questions. As I mentioned at the beginning, this is being recorded and it should be up in a couple of weeks. So look for that on our website. And please, everyone, take care and have a good day.